Hello everyone, we're the group discussing co-immunoprecipitation. So now the purpose. Co-immunoprecipitation is a popular technique that is used in cell biology quite often. The most important, or maybe should I say most direct use of co-immunoprecipitation is identifying physiological relevant protein-protein interactions. By using target protein-specific antibodies, co-immunoprecipitation can indirectly capture proteins that are bound to that specific target protein. These protein complexes can be analyzed to identify new binding partners, binding affinities, the kinetics of binding, and the function of the target protein. I think it is also very important to note that co-immunoprecipitation can be used for identifying physiologically relevant protein-protein complexes. Because this technique can use lyse cells and non-denaturing conditions, it can give a glimpse of the actual protein-protein interactions that are happening in vivo or in the cells.
Co-immunoprecipitation was performed in order to determine which proteins interact in the COVID virus cells. The proteins in question were the S proteins or spike proteins, the N proteins, otherwise known as nucleocapsid proteins, and the M proteins or membrane proteins. However, for the sake of this explanation, we will only discuss proteins S and N. The antibodies for each of these proteins were needed in order to assess whether they interact or not. The first step is to lyse the cell in order to gain access to the proteins and use a buffer. The second step is to add magnetic beads that are linked to antibodies against one of the proteins. There are other substrates to which the antibody could be bound and other methods to separate the proteins. But for our sake, we are going to go with the example of magnetic beads. Let's say an antibody against protein S, which would be linked with the magnetic bead. The anti-S antibody binds to protein S. The diagram is demonstrating the situation where S and N are not interacting. Then we centrifuge the sample, which pulls down the magnetic bead and in turn pulls down protein S. In the case where protein S interacts with protein N, both proteins will be pulled down to the bottom of the fraction. If protein S does not interact with protein N, then protein N would remain at the top of the fraction. Again, this image depicts the situation where protein N and protein S don't interact. The next step is to collect the pulled down fraction, which is at the bottom of the centrifuge tube. This fraction contains protein S and the anti-S antibody, and possibly protein N. This fraction is loaded onto the STS page. STS page separates proteins based on their molecular weight. A sample that contains all the proteins is also needed and is loaded onto the STS page. This would act as a reference for where the bands would appear on the STS page for each protein in question. After the STS page, a Western blot was performed in order to visualize the bands that represent the proteins. The anti-N antibody was added to the pull-down fraction. In the Western blot, if protein S and protein N interact, then protein N would be present in the sample that was added to the STS page, and the anti-N antibody would interact with protein N. Then there would be a protein band present that represents protein N. However, there would not be a band that represents protein N if there was no reaction between protein N and protein S. If this explanation didn't fill in all the blanks, hopefully next, our analogy should solidify your understanding.
Okay, so I'd love everyone to close your eyes and imagine for a second that we are at a dance competition. If it helps, maybe think about the movie Grease. There's an old-timey dance competition where we are all dancing in pairs. And you and your pair always have to stick together, no matter what. Suddenly, the announcer tells everyone to come up to the stage if you are wearing green. You look down and you see that you are wearing green, but your partner isn't. So, what are you going to do? Of course, you have to go up to the stage because you and your partner have to stick together. Once you get to the stage, the announcer can see who you are paired up with. And here we have co-immunoprecipitation. The announcer in this situation is like the antibody. You, the person wearing green, is like the target antigen. And finally, your partner is the other protein you are interacting with. Overall, in this quote unquote dance competition, the announcer is able to see person to person interactions, just like how in co-immunoprecipitation, the antibody is able to uncover protein to protein interactions. So hopefully this can help you understand co-immunoprecipitation a little bit better.
The question asked in this paper was what protein-protein interactions are occurring in MERS coronavirus and SARS coronavirus. They studied both to determine whether results obtained from MERS can be applied to SARS. The hypothesis was that there were novel protein-protein interactions in MERS and SARS that can be targeted to develop strategies against virus infection. Protein-protein interactions with spike proteins in cells infected with MERS were identified through co-immunoprecipitation. First, antibodies against MERS S and M proteins, seen in the figure colored pink and green, were bound to magnetic beads. Then, co-immunoprecipitated proteins were found using Western blotting, using antibodies for MERS S, M, and N proteins, as well as antibodies for SARS S protein and E protein. N proteins are the yellow-colored spiral in the figure, and the E proteins are the vibrant yellow proteins in the cell envelope. The proteins first isolated by antibodies and centrifugation were the MERS S and M proteins. Then antibodies identifying MERS S, M, and E proteins, as well as antibodies identifying SARS S and E proteins, were added. Through a Western blot, the proteins attached to the MERS S and M proteins were identified. This addressed which proteins were interacting with each other.
The big picture takeaway is that researchers are able to target the interaction between the S and N proteins in MERS and SARS, and that this could be a possible treatment method. There were antibodies that were against S proteins, M proteins, and N proteins used. However, there was no M proteins in the immunocomplexes that precipitated with the antibody for protein S, which meant that there was not an interaction between the S and M proteins. The N protein precipitated when the antibody for protein S was used. This demonstrated that they were interacting. The N protein expression increased rapidly during the 72 hours for Western blotting, more so than the M or S proteins after the onset of the infection. Looking at the figure, the N protein band was too intense. So further research and methods needed to be done past this specific test because it was difficult to confirm the presence of the S and M proteins in the immunocomplexes.
Thank you for listening to our presentation. We hope you enjoyed.